Welcome. In this lecture, we will talk about anatomy of dicot roots. Stem and leaf dicot we would understand in a separate lecture. Monocot roots we have already covered in a different lecture. So now coming on to a dicot root structure. So this is a TS, a transverse section of a dicot root. Here, epiblema is the outermost layer. Epiblema is also known as piliferous layer. Why? Because it has openings through which root hairs are seen, and these root hairs provide water and nutrient supply. Now these epiblema cells are relatively smaller in size. They are well arranged and mainly parenchymatic in nature. Again, the next layer below it is the cortex, which is again a parenchyma layer. But here we do have intercellular space that is present, and this is the part where starch is actually stored. Next to it is endodermis. Endodermis we can say is the innermost layer of the cortex, and here there are interesting stripes which are known as Casparian stripes that are present. These actually prevent the plasmolysis of endodermis, and they do not allow the passage of movement between the endodermis and the pericycle. So again, a very important aspect of the Casparian stripes. These are bands of thickening which run either tangentially or radially to the wall. Next layer is pith. Now pith is a layer which is found in the center. Now this center is the pith. In dicots, it could be present, it could be absent. If it is present, it is again a parenchyma cell, but here there are no intercellular spaces that are present. However, in the case of cortex, there was intercellular space that was present. So pith does not have intercellular space. Coming on next is the conjunctive tissue. Conjunctive tissue is actually the separation of the xylem phloem bundle from each other through the thin walled cells which are known as parenchyma, and they become meristematic to form the vascular cambium. Now we do have vascular strand. Vascular strand is a composition of xylem and phloem. Now here the phloem, the xylem, protoxylem is outside and metaxylem is towards the center. Since this is the case, we call this as exarch. Now note, in the case of dicot stem, it is endarch. Protoxylem is towards the center. Here, in the case of dicot root, it is exarch in nature. Vascular bundle is radial in shape that we can see, and protoxylem is away from the metaxylem, from the center. So metaxylem towards the center, protoxylem outside the center, and therefore we call it as exarch. Now if there are 3 xylem bundles, we call it triarch, 4 we call it tetrarch, 6 we call it hexarch, 5 we call it pentarch. So based on number of xylem bundles, we call it by different names: pentarch, hexarch, and so on. The next is pericycle. Pericycle is the single layer which is seen below the epidermis, and this is the outermost boundary of the stele, and the lateral roots whatsoever are present, or we can say penetrate, from the pericycle. Now another important thing that we must understand is the secondary growth that takes place in the case of dicot root. So in the case of dicot root, we need to understand that dicot cambium develops at the time of secondary growth. Now when there is a cambium that develops, the cork cambium is actually a result of tangential division of the outer cells of the pericycle. So these pericycle cells actually divide, and that division leads to cork cambium. And this cork cambium is similar to what is found in the dicot stem and is called as cork cells on the outside and parenchyma cells on the inside. So cork cells on the outside and parenchyma cells on the inner side. Clear? So this is a quick update about the anatomy of a dicot root. Next we would understand dicot leaf and dicot stem as well. Stay tuned. Thank you.